biology has a really strong role to play, not just in the way that a lot of people think. Like people think when you're putting biological product on the soil, it's just going to, whether it be break down organic matter or change the soil structure or what have you, it can play a role in so many different areas.
Welcome back to another episode of the From the Soil Up podcast. Today's special guest is Paul Daly from EMNZ. I'm pretty stoked to have a chat with Paul today because I personally use EM with some of my fertiliser recommendations, but I called him up the other week to run past a few scenarios that I thought that'd be really beneficial to use with, and he was great in giving me some of his time to basically say that I can use it for quite a range of solutions that I'm working through at the moment. So, Paul, why is biology so critical for productive and resilient farming systems?
First of all, hi guys. Thanks for having me, Conan. To answer your question, I think it is often the neglected facet. And while the chemistry side of soil and fertiliser often gets the press and the budgetary spend, probably more importantly, the biological side is every bit, if not more important, to how that plant responds, grows, and can thrive in any given environment, given that the biological element of the soil is the pathway for any nutrition getting to the plant, and also the host of that plant's that plant's environment, essentially. So it houses the biology, and it houses it obviously facilitates getting moisture and everything that is critical for that plant to survive. So that is why biology is so important, and it is often neglected, and that is the reason why we often see dire situations in some of our farming ecosystems.
Absolutely. So for those who aren't, quite familiar with what biology actually is, because it's quite a broad term, how would you describe it in practical terms?
Yes, I guess it's the living part of the soil that is responsible for so many crucial parts of its function, and also the food chain within humanity. And so what that living part of the soil is essentially made up of is microscopic living organisms, whether it be bacteria, fungi, protozoa, right through to your earthworms. And they are a thriving ecosystem that functions synchronously together to essentially create a healthy soil and create a healthy environment that enables plants and diverse ecosystems within the soil to thrive.
Awesome. So, Paul, for those who aren't familiar with who you are, can you give us a bit of a background and introduce yourself?
Yeah, so my name's Paul Daly. I am a director at EMNZ, or Nature Farm is our actual business name, but we trade under EMNZ, and that's probably the name that most people will be familiar with. But we are a company that manufactures microbial solutions. So that's a fancy way of saying we manufacture EM, and we also put that into different biofertilizers. So by the definition of biofertilizers, and how I'm speaking about it, is biological substances mixed with living organisms to help to elicit a soil or plant response when applied. So how that looks is we use EM as our biological source, and then we have relationships with a whole host of different biological companies that we use their ingredients to manufacture different things. It hasn't always been like that, and I guess I'll get into a bit of history about the company. It sort of has been a long journey. My father, Mike, he was an agricultural scientist, a field technician. He worked for originally MAF, and then on to AgResearch when the DSIR split up. I was born at Tara Hills, so that was one of their stations in the Mackenzie country. Not long after I was born, he was moved to Winchmore, which I'm sure a lot of people will be aware of, which is a research station in mid-Canterbury. And I think there he started to get into a lot of organics research. So he was doing a lot on they had an enviable organics research, like world regarded in the 80s. And unfortunately, that was completely destroyed when they disbanded it, because when they broke up the Crown Research Institutes, AgResearch got some land, crop and food got some other land, and it was all parceled up. And then also, all of the researchers then had to compete against each other for funding for those areas. So all the funding sort of went out of organics, and then once they put any ag chem on those previous organic blocks, it completely ruins any sort of data they can get off those. So it sort of that sort of went to the side. But Dad went to ag research, and he continued to do research, but on different things. But I think the seed was planted in the organic area for where he had a lot of passion and interest. And he was at a conference in Brazil in 91, it's either 91 or 93, and he met Professor Higa, who was the creator of EM. And they got talking, and they invited him to bring it back to New Zealand to research. Obviously, at the time, he was working for ag research. And so he brought it back here and did a lot of work at Harts Creek Farm. So Tim Chamberlain, out of Leaston, pioneer in organics. And so did a lot of work there looking at cereals, and also they did corn, onions, peas, pasture. And so he's got a couple of papers that were written out of the research that he did at Harts Creek. That continued on for about 4 or 5 years, so quite a long period of time. And then they identified that there was something to the product, like they were getting reasonably good results. Obviously, at the time, you're talking about sort of mid-90s, the knowledge and information about biology and biological systems and how they function within soil was probably a fair bit more limited to what we're doing now. Like I'm sure it was quite difficult to get farmers on board to think about using it. But at that time, he wasn't selling it. Obviously, he was just trying to get research done. And so following that research being done, ag research, he went to ag research. They published the results, but then ag research's partners. So how it worked at that time is ag research would research technology, and then one of their partners, like for example, like Ravensdown, was obviously take that technology on and start using it. But the way EM was set up at the time was it was very philanthropic, and you had to put a certain amount of revenue into a not-for-profit to produce research, and so that didn't really align with the commercial elements of ag research's partners. So Mike asked if he could do it on the side, and that sort of side gig went from 1 day to 2 days, dropping 1 day to 2 days at ag research, and then he asked them to drop a third, and they said no. So he went out on his own. It's sort of already at this time was sort of my first introduction to EM, sort of late 90s, I guess. I was still high school, but I remember Dad used to have tanks in the garage where he'd be making it, and I used to have to ferry hot water from the bathtub to the garage. And I tell you, it was a fair workout on the shoulders. But that was sort of my first introduction, lugging water around for in order to dissolve the molasses so that fermentation could occur. But getting back onto the from there, Dad sort of did a wee bit on the side. He was doing consulting, and then he started the business in 2001. And so I'll give everyone a guidance of what EM is. Essentially, EM's a multi-species, multi-family microbial brew. It's made up of bacteria, fungi, and yeast. And these microbes sort of sit and function with synchronicity together in the soil to help boost. So first of all, it was designed to digest organic matter. That's what Professor Higa found out. Created it for back in the late 70s. But it's also very good at stimulating native microbes, and also other function along, helping the plant absorb nutrients. It's great at outcompeting disease and that type of thing. And actually a really good story, sorry for jumping around, but a really good story Dad always tells me is what happened was when Higa was developing the product, he was he actually had a chemistry degree, and he was working with ag chemicals. This was back in the 60s, I think, and he got really sick, and so he wanted to change course. And so he went back and studied, and he always remembered his grandfather utilizing lactic acid bacteria to deodorize dung and break down straw that had animals on it and that type of thing. And it got him thinking that, how can we and I utilize microbes to enhance production and enhance an environment. But his thought was, how can we put multiple families together? Because if one works well, then multiple are going to work really well. And so that's where his study really came in. He started to look at, really under the microscope, was look at how these microbes work together and whether they survive and what their behavior was. And anyway, at the end of each day, he was sort of putting the excess of the brews that he brewed up into a bucket, and then he'd throw it out on a patch of grass outside of his lab. And then on walking in one day, he discovered that one area had like a huge amount of growth, and he was like, okay, hang on here. There must be something to that one. And so by a process of going back through what he had done, he managed to figure out that there were a couple of these species that were working really well together, and that was the foundation of EM. And so he then obviously worked on it for a long time, coming up with exactly how it works together. But that was the basis of it and the foundation of it. And I think one of the things we've noticed, and it sort of leads me back to where we are in the timeline of the business. But, there's when you're working with multi-species or a lot of different microbes, you get a lot of different potential functions. They can do a lot of different things. And that's been like a blessing for the product, but it's also been a curse in a lot of ways, because people are like, well, it can do, like, it's really good at breaking down organic matter, but it's also really good at outcompeting disease. But then you can put it in your septic tank, and people are like, wow, like in our day, in our society where, you have a problem, you go to Mitre 10, you pick something up, you fix it, and then the next day you have a different problem. That mindset can be difficult for people to understand, but it's really one of the benefits of the technology. And when you explain it, that's got a lot of different microbes, and not all of them are working in each different instance, but the function of one will kickstart the function of the others. It sort of starts to make a lot more sense. And one of the reasons why we see multiple impacts and a lot of different benefits when people are using it in different aspects of the soil and in the environment.
So that,
yeah, and so you go.
Yeah, so no, it's good. It's given us a really good overview of how EM came to fruition and actually what EM is, because some people won't be familiar at all. So very much like, I guess, working in a farm system, diversity is good even in the microbes. So when essentially there are a lot of microbial or biological products out there, but maybe they typically focus on doing one part of it. You're saying that actually having that diversity in there is more beneficial for the outcomes you may be looking for.
Absolutely. Yeah. Because, as I sort of said before, like certain microbes require certain conditions for them to thrive and to function really well. And if you don't have that condition, then if you're putting on a single species, it's either going to sit dormant, and then attrition, you're going to get them dying over time before they can function or thrive. Whereas if you've got a multi-species brew, when you put it on, one of them is going to find an optimal zone for it to start to function, and that process, what we've seen, that process will kickstart the others. And a good example of that is sort of organic matter breakdown. Like, it's such a big part of what the company was built on. And, in the beginning, when Mike started the business and even when I first came back into it, we'd actively ask people, how much organic matter you got in the soil, because we would want to ensure that there was organic matter there for the microbes to function and to have food source. But we've mitigated that over time by adding food sources into our brews to make sure they have the kickstart. So we've sort of learned a lot, and that's probably been a big sort of, we can help give this product a really good kickstart by doing this, and this, and not having to rely on a certain percentage of organic matter in the soil so that the microbes have enough food source to function. That's been sort of a big understanding for us. And I think, early days, and this will help me continue on with the timeline for you, but like Mike, when he first started the business, there was a market in organics for wine. Wine's always had a good market, and there's been a premium. So we've always had a good following in viticulture, and also like they have, they've always been conscious of having a good understory function. So microbial function's always, and dealing with fermentation, it's sort of always been a real easy sell for us. But, we're putting EM onto an understory, you really get a lot of bite. There's good breakdown. You get a lot of function. You can sort of, you see a lot of benefit. So wine was an early one, but, it was a pretty hard trek, I'm sure. I've talked to Mike about it in the early days, talking to dairy farmers or something and trying to explain biology in the early 2000s would have been pretty brutal. Cropping would have been a bit easier. But yeah, it was certainly something that was very fringe, I guess is the easy way to put it. And so I came back into the business in 2013, and I was always really interested in it. I did a paper at uni. Because I studied business. Mike, my father's a scientist, so it's a family business. But I did a paper looking at, and we had to do a business study, and so I looked at EM because Dad started the business, and I sort of deep dived on it over the course of a semester, and I really developed a lot of interest in it then, and really thought, oh look, this could be really cool the way the world's going. Back then, this is sort of 25 years ago, I probably would have thought it would progress further than it has now, but that's a discussion for a different day. But yeah, so I was always interested. And then when I was living in Melbourne at the time, and we had just had our first child, and Dad came over because he's always travelled a bit because EM's obviously all around the world. We have really good relationship with the European and the American and the Asian partners. Specifically Asia, we're sort of in that zone, but we spend a lot of time talking to the Europeans because their systems are way more similar to us than what the tropical systems are and how they use the product. But yeah, so he came over on the way home from a trip and he sort of said, oh, well, if you're thinking about coming home, because we'd sort of talked about it, why don't you come to business with me? And it was a fairly big change because I was working in a big events company and doing commerce stuff. And so it was a bit of a leap of faith. And at the start, we had a few distributors and we were selling a lot direct, but we were really tied to the dairy industry. Like a couple of our distributors were solely dairy focused, and then sort of within a year of me coming home, that big dairy downturn hit in late 14, early 15, and it really had a dramatic effect on our business. There's a lot of lessons to be learned. We had our eggs in one or two baskets. We're sort of geared towards a distributor of 50%, and when you that's really risky. So out of that came a lot of opportunity. Like Mike and I went over to Europe in 2014, and we went to visit some European partners, and they were doing some really cool stuff with EM, like a lot. And and there were restrictions already in the Netherlands where they weren't allowed to use a lot of N and especially P, and they were doing a lot with effluent and injecting it under the ground, and then also a lot in fermenting organic matter, so what we call a bokashi, using effluent with plant matter and fermenting that, and then so quite innovative ways. But then actually when we went to Switzerland, they were fermenting it into sort of like a chip, and then that was being used as sort of a amendment to soil. So it was sort of a solid way to put it on. So that gave Mike and I a really good idea because we were already working with a number of FERC companies here, smaller guys like the biological guys, like sort of your main and minerals stuff, and we'd sort of met and some real pioneers, I guess, of that early the biological industry sort of 10, 15 years ago. So we'd started to work with some of these guys, and we had an idea that, in New Zealand, not a lot of products are actually sprayed on, really, in the scheme of things. Most of it's supplied granulated. So we were like, okay, well, we could come up with a granulated form of EM. And we'd found, in sort of early days, we were really like discovering different ways that EM was helping people. Like there were liquid fert guys that were putting it on with fertilizer, and that anecdotally we were getting a lot of feedback to say when we add this in, we're getting really good response. So it's helping the plant to absorb it and stimulate that biological function. So So really early days, we were using EM for system change within the soil. So you're breaking down organic matter, you're stimulating all of that microbial activity, and then you're helping, you're getting earthworm activity, and you're really seeing a system change within the soil. And then people would put other things on, and sort of we were there to do that, and we were a bit of an afterthought everywhere else. But we started to think, okay, well, what are other ways that we could be influencing a wider area of land within New Zealand? And one of them was coming up with a solid product. So when we were in Switzerland, we sort of got the idea, and we come back here, and over the course of sort of like a year, we started to look at different materials that we could essentially grow EM out on. So how we grow EM, essentially, like we have a license with Japan. They create the cultures, and then we brew EM1 up from the cultures. And then what you have to do is you have to take it a second generation. And so that second generation is essentially you're just putting molasses, water, and EM1, the concentrate, and brewing that up in an anaerobic environment in a big wine tank. But this granulated process, essentially, we'd be doing the same thing, but instead of using water as a substrate, we'd be using a solid material. So we were using bentonite, we were trying different things, and we came up with zeolite. We found some zeolite, and that works brilliantly. And essentially, I won't go into too much detail because I don't want to give away the Colonel's secret herbs and spices. But we created, we put a couple of solid food sources in there, and zeolite, and a bit of water and EM and molasses, and we fermented it all together. And then we bag it, and that became our product, Fert Enhance. And it doesn't have to be used with fertilizer, but we wanted to really get targeted with the branding, so people could understand what they use it for. And so essentially, and it needed something to be blended with, because you can't really spin it because it's about 22% moisture content, so it will bridge quite easily. So it started to get used, and this is sort of around the time we met Rob Flynn. We have a great relationship with him, and he really started to push it out. And then we got some good relationships up in the North Island with, your Evokers, your Kiwi Fertilizers, your Dicky Direct is a bit later than that, but these companies really started to push it out within Fert mixes, and so we really started to expand our area of use, and we saw some fantastic responses, because you're putting the biology right beside fertilizer, and admittedly these guys are using pretty soft fertilizer. It was like guano and things that are probably a lot more biologically friendly, and Evoca use dicalcic. But it's still been used with super and other things, and in actual fact that doesn't have too much of an impact because EM's quite an acidic product. But yeah, we started to understand its effectiveness in a lot of different areas, and that was sort of a key evolution for the business, and also probably a key evolution to our understanding around products and creating things that work for the grower, rather than having one product. But before that, we just sort of had EM, what we call EM ready to use, or people could buy it in the concentrated form and then do the brew up on farm. So we started to get a bit more targeted and a bit more, and that was great for growers because they could understand a bit more about how it's used and how to utilize it.
Yeah. So I guess, like, you've been in the business for quite some time now. Was that evolution to the granular almost to try and take away a little bit of that barrier, because there isn't a lot of soluble or liquid fert going on?
Yeah, 100%. And sort of following that, we sort of started to get a lot of traction, and then Flinny introduced us to a whole new market. It was sort of like what I like to call the biological regen market, and we sort of met Nicole, who's been great, like Cheryl Prue. I don't know if you know Cheryl Prue, but she was Soil Food Web back in the day, and then like people like Jules Matthews and John O'Frew were sort of crucial in probably the next evolution, because they're very cluey in biological function and also just in what's the farmer needs, and the farmer is going to be beneficial to them. And so from there, we sort of created a couple of different products. We've got a product balance, which is fish, amino acid, humic acid, and EM. And that was really derived from, there's a lot of a burst in understanding around fungal activity in soils and the crucial aspect that fungal activity creates. And so EM's quite, it's got bacteria and fungi, but it will play pretty neutral in the soil. So it'll boost everything, but it's not going to increase the balance too much. If something's out of whack, it will really try and change it. But there's a lot of at that time, these guys, especially Nicole, were talking a lot about bacterial fungal ratios. And obviously, us doing work in tree crops, we were sort of trying to influence that. And so, with balance, we really tried to look at how EM can dramatically have a fast impact on fungal populations, mycorrhizal and that type of thing. And so, through the doing soil food web testing, we figured out that, and through a lot of these guys' knowledge, that fish and humic acid are really good fungal boosters. So by pairing it with EM, we turn on that switch essentially, and it has a dramatic impact in fungal activity, and also like our digestion speed went through the roof, because you know what it's like when you're looking at digestion in the soil, your bacteria will go after your simple sugars and start to break down the easy organic matter essentially. But then when you get into your lignin and more complex organic matter, you need your fungi in there to break that down. You need to stimulate your earthworms. So once we started pairing some of these products together, we started to see like a dramatic change in digestion and also overall activity. So we sort of started to see a lot of benefit for growers, and that's just coming from our understanding and working with these people that had really good knowledge in these different areas. Yeah. And so that's been super beneficial to us as a business, and also to me when coming up with products that are going to service and offer value to customers. Because at the end of the day, we sort of touched on it earlier, but in New Zealand it's all about yield, but really it should be around like value. And so it's our job to add value to growers, or not just growers. We as I'm sure we'll touch on, we have products in the waste industry as well, and also we've got quite a big following in the turf industry, and they're more about aesthetics and everything looking good, because but it's all about adding value, and what we found through that time is we really got to understand what value EM was adding, and then if we bolt these things into it, we can really add a significantly more amount of value. And I think we started working with a number of companies around the country that were probably considered competition prior to, but now they're customers of ours, and we sort of have a really good relationship with them. And I don't mind naming them, like Biomarinas, for example, and Waikato and Southern Humates. We have good relationships with them, and oftentimes businesses they're trying to keep the customer to themselves, or they don't want to let any other business in. But through collaboration, it can make everything so much stronger and so much better. And I think we've found with those guys, and also I think how I met John O'Frew was he was working for Tim Chamberlain at Harts Creek, and they were using different organic substances along with they've been long time users of EM. Obviously that's where we did the trials, so using it for the last 25 years, but they're also using seaweeds and fish and different type of things to enhance crop nutrition and crop health. And so we sort of started playing around there, and we did some work that they grow barley and wheat leaf and dry it for a supplement industry. So we looked at barley and wheat leaf, and we were looking at seaweed and fish plus and minus EM, and we saw a dramatic difference. And this is where it's sort of a light bulb moment of like, okay, we can add these things and it can have a huge benefit for the grower. And oftentimes, I will tell the grower, like, for example, like if we're putting EM on, we might say 10 litres a hectare or 20 litres a hectare. Over the course of the season, you can use 30 litres. But if you're using some of these other biological products, you can pull our rate back, just like you can pull their rate back, because they'll make each other better. Because at the end of the day, it would be short-sighted of me to say, no, you've got to keep putting your 10 litres on, because at some stage the farmer's going to come up with a budgetary constraint. He's going to start dropping products, and who knows what that'll be, and one of the days it'll as I said, it'll probably catch up. But if you can impart a bit of wisdom and knowledge to a grower and say, look, these products work really well with ours, and you can use a bit less of each and you'll get a really good response, that's to the benefit of the grower. So Yeah, we've got a number of products. A lot of our products actually have these other biological ingredients in it, and we've seen a great response for the grower, and also it's been good to have those connections.
Yeah, and I think that's a really important point you've just raised is like being actually working together and collaborating for the benefit of the grower or the farmers. That's actually builds more trust within the
Yes.
market. I guess biology has probably been historically quite undervalued in agricultural systems. Why do you think that is? Because it might be one of the first things that gets dropped out of, like you say, the budget when things get tight because the farmer might not fully understand its role.
Absolutely. One, I think it's out of sight, out of mind, right? You can't see it. And so, unless you're, and I guess the advice I'll give any grower out there is start looking, get a spade, go and count earthworms. That was like in the early days, as I said, like one of the things that I did was going out with all of these guys. When I first came to the business, we sort of had a couple of decisions. It was, do we start a fertilizer company and use EM with it to try and get it out there, or do we try and become an expert in one particular area? And that's what we've done. We tried to be an expert in EM and biology now. And we sort of, I like to say, we've taken our wheels off as our training wheels off as a biotech company over the last few years. But I'll get into that in a minute. But yeah, I think you really need to be looking, and so yeah, earthworm counts, and I always encourage distributors to do that because and also growers to do that because it's a really easy way to understand when something is impacting what's happening below the soil, whether that be amendments that you put on or whether it be management practices or the climate. So it's a really easy way to do that. There's also tests you can do, simple ones are like microbiometers, but also you can do soil food web tests. But also, it's different than fertilizer. Like people automatically have the fertilizer hat on when they think about anything that they're spraying on. And so when putting biology into a system, they expect a fertilizer response. And that's been a challenge that we've had since day one, if I'm honest. And I sort of spoke to you earlier, we've got one of the products we're working with, our plant stem product, probably has that sort of response now, and it's sort of as in it boosts growth. But like EM balance, like all of our other products, and even all of the products that we work with, the feeders, they like yield and growth is a secondary function, and I like to try and explain that to growers. Primarily we're looking for system change within the soil. We're looking for a healthy soil where we can. It's got a really nice earthy smell. We're looking, to when we open the soil up to see good aggregation. We're looking to see earthworms, and then we're looking to see healthy plants like less disease, all of that type of stuff. And then after that, we're going to see a growth response for the plant, but they spray it on or put it on, they expect to see a growth response. And that's that just comes from historically what we see from application to fertilizer. And so I think it's starting to change, but it's still very much there, and it's still something that we're like a challenge that we're always up against and have to educate on out in the market. But yeah, it's sort of a reality, unfortunately. But what I would encourage people to really start looking under the soil. We see huge impact under the soil far faster than we see impact on top of the soil when it comes to the products that we're putting in for system change, which is like our main EM products.
Yeah, exactly. Normally you see it like I like to think about it as when you use it as like almost when you're coating the seed. And people think
Yes.
they don't see much happening above ground, but below ground there's this huge root ball that's growing, and then it'll just rocket off. For those,
Yeah.
you probably get quite a few farmers that are probably looking to use, I guess, less synthetic fertilizer products. How does
Yes.
biological tools actually help them, I guess, maintain that productivity? You probably alluded to it a little bit just before.
Yeah, I think first of all, it's a multifaceted approach. So first of all, you're boosting the function of biological activity in the soil, and through that, you're enabling the plant to get access to more nutrients. So what available nutrients are in there, and so and also you're digesting organic matter, which is going to make plant available nutrients get absorbed by the plant. So there's multifaceted ways in which biology will do that, and it's a pretty crucial process. But if you want to go deeper, obviously the pathway between the plant and getting access to anything is very important as a biological pathway, and it's a pretty crucial pathway that's happening within the soil. You've got quorum sensing, where a plant's telling the microbes what it needs, and they will pass that through to the plant. So if you can fire up the activity of that within the soil, or supercharge that, then you're going to get more nutrition heading through to the plant, and the plant's going to get access to more crucial elements that it needs. It's going to have a larger root area, so you're going to get access to more moisture. So and also that biological activity is creating a healthier soil environment, which means more macropores, more airflow, which all points through to a healthier plant and a better growth potential for that plant. And so, it's pretty crucial in that. And we sort of saw that really early on, was whenever we put EM with nutrients, everything would work, or the plant would grow a lot more effectively and a lot healthier. And so we started to play around really early with it. Mike did a lot of work early on, and our early fruit enhanced trials are all sort of half rates of MPKS and getting a higher growth response. And that's all just biology pushing that through to the plant. There's probably a wee bit more at play as well. One of the things that we have discovered, done a lot of work on, is EM creates a lot of organic acids, enzymes that are really good growth promoters in themselves, but they're also really good at things like stimulating stomal opening, and also stimulating absorption through roots. So you're getting more uptake either way, and then that leads to more nutrient getting in, if you're pairing it with nutrient, that is. But if you're just putting it on the soil and it's been regularly fertilized, you're going to get more uptake as well, because those microbes are going to go and break those nutrient compounds down and pass that through to the plant. And like really interesting things. There's so much data out there, and I think we're really lucky in that EM's like used globally around the world. There's so much research data on it, and as I've sort of started to understand a lot more, I've been able to search specifically for things and find great data. Like one of the things we discovered, it probably would have been 5 years ago, but like EM production in the soil will produce phosphatase, which phosphatase helps to unlock bound up phosphate. So we always knew that. We would see that by putting EM on, plants will grow really well, and they get access to phosphate, or we'd see phosphate levels rise, but we never really knew the mechanism. And now we're starting to understand, it releases this enzyme which helps release bound up phosphate. But that's not just exclusive to EM. There's lots of enzymes that do it. We're actually we've bought some precision fermentation equipment, and for the last sort of 2 years, we've been really honing in on looking at specific microbes that can help to really boost production in different areas. So we've looked at a Pseudomonas species, which is really good at unlocking bound up phosphate. We're growing an Azotobacter species, which is great at nitrogen fixation. And we've also done a bit of work with Bacillus, which is a really powerful biofungicide. So it's great on powdery and fusarium and a lot of other things. But it's actually already in the market with like Bayer's got a Serenade product like that. There's some, and so one of the great things is to see is that some of these larger multinationals are actually looking at biology and starting to promote it. I will add, after years of downplaying it, but it's good for the market. But we've sort of started to look really deeply at that. The funny thing is when we do these precision, we've spent hundreds of thousands of dollars on this equipment, like the really fancy bioreactors, and we've got these high-tech nitrogen fixation products and phosphate solubilizers, and then we throw them into the market, into field trials, and EM still outcompetes it a lot of the time. So we're still sort of honing in on that area, but it's sort of it's given us a real appreciation for EM in that regard. But I think it's because it's, again, we'll touch on it's multi-family, multifaceted, so it works no matter what. Whereas we need specific environments for these other things to work. So that's where I think that sort of comes from. But yeah, it's been a real learning curve for our business. We're working with a microbiologist from Auckland, and we've got a biochemist now in the business, and we're really looking closely at that. But the ease at which we can make EM, because these other microbes, you're fermenting them in, as I said, bioreactors, which require precision pressure, agitation, precision temperature, bubbling, airflow. It's really a delicate process. They don't like any competition. And then here we have EM, which we brew in 10,000 litre wine vats. We put molasses in, which not sterilised molasses, just molasses, blackstrap molasses. We put the EM1 culture, water. We fire the heat into it, and it just goes for it. And that's one of its real strengths. And it's actually that process that enables it to be so easy to brew, because the lactic acid, because it's made up, EM's made up of, as I mentioned, bacteria, fungi, yeast, but it's lactic acid bacteria, photosynthetic bacteria, actinomyces, fermentive fungi, and sort of Aspergillus, Saccharomyces, these type of species. And what makes it so great to grow and also so robust is when you put the molasses in, the lactic acid bacteria goes crazy, and it produces lactic acid, and that drops the pH, and that immediately creates an environment where nothing else wants to grow. And so it creates its own stability, and then the other products and microbes are the only ones that are going to grow, because they'll be put in there and they grow throughout that fermentation. And it's a really neat way to do things, and it's given us, and this new process, these new fermentation tools really given us a great appreciation for EM and how to do it.
Yeah, that's bloody awesome. I'm looking forward to seeing what you develop over the coming years. So if we think about it, some kind of specific examples that I'm personally interested in utilising it for.
Yeah.
Obviously, effluent ponds, quite a lot of people feel like it's a waste product. Like, is EM's a product that we can utilise here to kind of change that, I guess, the form from a kind of anaerobic to an aerobic, and it has that effect, like you're almost bubbling it up and utilising it out through like a pivot or a effluent or a shittigator or whatever?
Yeah. It won't really bubble it up. What it will do, so EM being facultative means it can operate in an anaerobic or aerobic environment. So that's why it's really good for compacted soils as well, because it will go in and open it up. But in an effluent environment, what it's really good at is eating crust, obviously, like surface crust, because it's all organic matter, right? Like whether it's in an effluent pond or it's in a soil, it's organic matter. So the EM will go in and start eating it. And more importantly, you get in, like what happens in an effluent pond is like the farmer will see surface crusting, but in actual fact, it's the material that is at the bottom that's the big issue, and that's inert, like it's a complete dead zone, and that's decreasing their capacity of their pond and also the function of their pond. And so what EM will do is it can actually go down and survive in that inert environment, because it can survive without oxygen, and it will start to digest that and bring it to the surface and break it down completely. And so what will happen over time is you'll see the surface crust go down, but more importantly the sub inert material is being broken down, and you'll get a flowing pond, so it will just be effluent. But more importantly, I guess, is you're creating a supercharged fertilizer almost. You're creating a highly available, microbial rich, nutrient dense liquid that is going to really benefit your plants and environment, because EM's going to grow, so you're going to get a huge amount of beneficial microbes going on. Another thing in an effluent environment, and it's in any environment when we see a lot of disease or a negative environment, when you open soil and you smell a lot of sulfur and that type of thing, it tends to be that the negative function or the negative microbes are really multiplying and dominating that environment. And so Michael always gives a really good example of this, but he says it's sort of like the microenvironment is sort of like like the political environment of any country, right? You sort of have your extremes, your left and rights, and then you have a huge floating mass, and they'll just go they'll go either side depending on what's better for them, what policies. And that's sort of like the microbial environment. You have really like your good microbes and you have bad microbes, and you have this floating mass that will sort of do the function of whatever is dominant within an environment. So where you have in effluent or in soil where you have a lot of negative function, it's because that mass has been tipped over your negative side. You get a lot of sulfur or ammonia smell. You get a disease forming. And by putting a product like EM in, a lot of healthy beneficial microbes, you can help shift that mass across. And they'll start breaking things down, and that gets the positive function happening, and so that environment can switch quite quickly. And we see that quite a bit, especially as I said, in compacted soils or in effluent ponds where you got a lot of crusting, all that that bottom material. We see it breaking down and we see it flowing a lot better, and then we see a really good response in the paddock. And so in effluent, we do that. We have I didn't really touch on it, but EM's used a lot around the world in waste, so wastewater, effluent, waterways, and that type of thing. We're using it quite extensively in waste here as well. We've got a lot of compost plants around the country, Living Earth here, but also up north that are using it to speed up the composting process. So the composting process, generally 12 weeks, they're seeing good results in 9 weeks with better breakdown and better microbial activity. And Living Earth, we're often seeing like full mycelium covering stacks and that type of thing. It's been really, really cool to see. And then also like Synlay's using our stuff to treat all their DAF waste down at the Dunsandel factory. That's been like for 4 years now, and they're having great results there. It's quite an interesting situation. They got all of these dairy farms around it connected with big stainless steel piping, and they're not allowed to flush those because their resource consent for water doesn't allow it, because it's like kilometers of piping, so it'll be a lot of water. So when they're flushing, then they're putting that DAF waste through when it gets to the irrigators, and then it stops off, might not get turned on for another 3 weeks. And so you can imagine these hot stainless steel pipes with DAF waste, like it's going to really ferment and putrefy. And so they were getting a lot of odor complaints, even from motorists down State Highway 1. But so they come to us, and we did a few trials. So we looked at how we generally work in a waste situation is we'll treat it at like 1 to 50,000 or 1 to 100,000 down to 1 to 10,000, and try to see where the level is that EM's having a dramatic impact on either breakdown of a waste material or odor, and then we'll sort of recommend a dose rate from there. And so we did that. We did some bucket testing, found a good impact at a certain dose rate. Then we did a month test on a trial, where we're pumping it through the lines, and they were getting different people who worked at the factory to walk out into the field afterwards and smell. And then they were they mapped complaints to ECAN, and they had a 90% reduction in complaints during the trial period. So they've gone full noise for the last 4 years, had really good impact. And then we're also seeing really cool things in like reduction of sodium in the soil and different things that we're seeing because the microbes are breaking that down, because there's excess stuff that's coming out of that DAF waste. Often when we're putting waste material on, we might see it in if on a meatworks or something where they're putting effluent or washing onto a field, we see adverse effects in nutrition or levels that are going to severely impact growth or palatability of that particular crop. So we see the microbes are really good at balancing that out, and we've seen that around the world in different things. Another really cool thing that EM being Japanese based, when the disaster of Fukushima happened, obviously a lot of farmland was radioactive or impacted with radioactive material. And so they started doing work with EM to look at, if you put EM into this environment, what's going to happen. And obviously microbes can't break down radioactive material, but what they started to see is that the microbes were stopping the plant from taking it up. And so that's that quorum sensing thing, right? So they're telling the plant that this material's bad and you can't take it up. And so in the control, they were getting radioactive material in plants, and in the the EM treated stuff, they weren't. So it was remarkable results, which is all published. So and they've been doing work since then. They've been doing work in Ukraine. So it's really like we're still scratching the surface of these fields, but the microbes can really have a dramatic impact in lots of different areas.
Yeah. I think another one that I'm pretty keen on trialing it in is like the hill country kind of sleepy soils. Like the aggregation's really good. They've got thatch build up, but the grazing management obviously can't stimulate things. So utilizing something like EM, which by the sounds of it will start breaking down that organic matter and getting those processes going and hopefully stimulating the biology that might be, asleep. Is that something you've come across?
Yeah, we've done a lot of work in hill country, and it was probably the granulated product that allowed us to do that, because it's a wee bit hard at times or expensive for a grower to spray it on in those sort of countries. So utilizing things like flying it on with lime or something like that was something that became available to us when we created the Fert Enhanced product, and it has a dramatic impact. I mean, I love lime anyway. Like, lime's amazing for growers. It creates the cities that our biology can live in. Essentially, it does so many different beneficial functions. But in this case, it was great because it's the perfect product to blend in with EM to get it on. And so we would see, we'd see really fast results because, as you say, there's a high amount of food source, and the microbes can just get to work really fast and really have a dramatic impact in that environment. And it's quite highly microbial, often quite fungal. You see a lot of fairy rings, fungal rings. But when you can really kickstart that environment with some biology from EM, the bacteria really kickstart it. It helps to redress that balance and have a dramatic impact on those sort of environments. We've done a lot of work in looking at pasture, obviously, over the years, and there's a good trial on an independent one done on our website, which we looked at pasture production. But one of the really cool things to come out of it was the impact of EM on clover, and that's just helping to create an environment where the clover can thrive. And another thing that lime will do really well is help to boost clover. But yeah, we often see a huge boost in clover growth after applying EM, and in a hill country environment, that's so crucial. You get a lot of, if you can get your sub established and booming, it can have a dramatic impact on the production in hill country. So that was something that was really cool to see bear out in an independent trial. I mean, we doubled clover production within that environment or within the sward, and then also in that one we looked at reducing urea, which also has a really good impact on helping your clover thrive because it's not getting shaded out. And that's I guess that also brings me to sort of like, in the early days we would anecdotally hear from growers, EM works really well at doing this, and then we would sort of like do a trial on it and would sort of figure, okay, it's doing really well, and then we'd and a lot of the time we were doing our own trial work because Mike's background in agri-research as a field technician really gave us the capacity to do that. And so we got some really good replicated trial work, but that will only take you so far. And so over the last sort of 6 years, we've made a huge investment in getting a lot of independent trial data, and that's really given our growers a lot of reassurance, I guess. And it's also helped us to professionalize our processes within our business from a research and development side of things. So we're always looking at different ways in which EM will have an impact, but we've now got a funnel in order to look at how it will work and then to get it to market. So I'll just give you an example. I mentioned earlier, we've got a new plant stem product, which is a fermentation extract derived product. So we use EM to ferment plant material and extract beneficial substances that enhance plant growth. And it's been really cool to this development process. But previously, I guess we would sort of be like, I mentioned John O'Frew earlier. He was like, fulvic acid. When we're using a bit of fulvic acid, why don't we put fulvic acid with EM? Because we're putting them together in spray tank. And so then we created EM fulvic because the market sort of said there was a thing there. And so we would sort of make a product reactionary to something else, basically start selling it and then research it as we were going. But we've sort of professionalized, as I said, our business a wee bit, and we've got more of a funnel. And so now we sort of figured out that there were a couple of really powerful growth substances that we were wanting to extract via certain plant materials, which I won't go into too much detail. But so we desktop fermented it, and then we figured out how we're going to large scale do it, and then we started researching it. And our process now is we always have a summer student comes and runs trials for us all summer. So generally a Lincoln student, and we do that through Callaghan, which has been great. And so we will look in the tunnel house. We've got a tunnel house here. We'll look at growth response, essentially, if we're looking at growth. And then we'll fine tune different, we'll look at rates, we'll look at timing. And then when we get a success, we'll then go and run our own field trials. At the same time, probably we've got a number of friendly farms close, and we'll get them to spray it on, get their feedback. We'll start to look, if it's soil, we'll start to dig, we'll start to get analysis done on what's impacting. So balance, for example, with that one, we were looking at digestion. So we were burying cotton pads and we were looking at crop residues. But like for Plantston, we then did our own field trials. And then from there, when we got successful ones and we knew what we were doing for particular crops, we then went to independent ones. And we did 3 years of independent trials on Plantston. And so we've got maize, we've got fodder beet, we've got pasture, we've got kale, we've got cereal. And so now we've gone to the market last year with it, and we've got this really robust data. And so it's really cool to see how the company's developed in the funnel to getting a product out and really getting good. I guess now the farming probably has a bit more reassurance that we know exactly what we're saying when it comes to rates, and we're not doing it on the fly as much. And when they put it on, they're going to get a certain response. But also that learning part, the 10 years of learning and producing different products and putting things side by side and doing it anecdotally, that was so crucial to getting us to where we are now for that understanding. But yeah, it's quite cool to see the evolution and that type of thing. But I guess from our perspective, we even though we're putting all the stuff in place, we're spending a lot more money, as you can imagine, on trials and that type of thing. We really want the foundation of our business is EM and putting things with EM to help boost that biology and to create an environment that enhances and produces healthy plants, essentially. But we want to do it. We want to make sure that it's really cost effective for the grower. So we really try to narrow our production process down and not to throw the price through the roof for the growers. So we're still really cost effective technology overall, and growers can still grow EM1, the concentrate, out on farm if they want to do that process. And that enables them to probably half the cost. So still really cost-effective technology if they want to use it out there. And that's the foundation of EM worldwide, really. They're wanting a maximum usage and a low-cost barrier to entry.
That's awesome. So that was probably going to be my next question is, I guess, farmers are going to ask, like, does this pay, like this biological stuff? And you've done all these trials. I guess there's a good consideration or probably feedback from some of those farmers involved in it.
I think early days, we probably got a lot of pressure from some of our larger distributors just to get more data so that they can put it out. And to their credit, when we've got the data, they've gone for it. Do you know what I mean? They've been like, okay, it's proven, we'll go and do it. But until we got there and we were able, and now we're able to put a dollar value to it, like EM might cost $30 a hectare or whatever, depending on the product. We don't tend to try and be too much over that mark for an application. And, growers might put a couple on a year, but we see a really good payback in terms of most of our products in the hundreds, if not thousands of dollars in terms of return per hectare for what they're getting, what they're putting on. And so there's a lot of reassurance around that now that we've got the data, I guess. But, it's a little bit harder in the soil products. For example, Balance and Soil and Crop are our two soil products where we're looking for system change because it's always a secondary function. But we're starting to do some really cool case studies around like digestion and things where they can see like that added value, and that's going to have a dramatic impact on different aspects of their farming system. So whether they are not burning off, whether they're not baling, whether they're just going to spray EM on to get that breakdown happening, and the return they're going to get from nutrient, especially K and things like that, are going to get breaking down. So we're able to put value to that. And also, there's also the secondary benefit of growth. So I think we're getting a lot smarter in that area, but just working closely with, consultants like yourself and other growers where we sort of get to understand challenges that the grower has and how we can impact them. For example, the other day I've just done two recent case studies, which have been really interesting. And one of them was looking at using EM alongside glyphosate. And we've had growers doing it for quite some time because for two reasons. One, you're creating a lot of dead matter, so you want to break it down. And two, the organic acids and things in EM help to increase uptake, believe it or not. But it's the same in foliar nutrition. We get really good stimulating stomal opening because of the organic acids in EM and also respiration on the leaf. So putting billions of microbes on the leaf, you're getting a lot of CO2 respiration, and so that'll open the plant stomata. So it'll impact it in several different ways, which is why we get great responses from a foliar perspective. But in this case study, we were looking at glyphosate. And one of the things is EM's quite acidic, so at a 3.5, you put 3% EM into a spray tank and it will drop it into that Goldilocks zone for glyphosate, which is sort of like your 4.5 to 5.5, I think, from memory, or somewhere around there. But a lot of obviously water's at 7, so you're not getting optimal zone. So, but that come from a grower just asking how can we and I was like, oh well, we can put citric acid in because we deal with a lot of different things. But it was great just to know that you could. And Flinny's been using EM for years for that, and I always thought the benefit was from the stomal opening and also the digestion. You're getting a better kill rate, but also a better breakdown on that dead matter. But it turns out that's probably got a role to play in reducing the pH of the spray tank. And then we also looked at kill rate using half rate and even a quarter rate of glyphosate versus and then a regrowth rate, and adding EM, and we got a dramatic reduction in regrowth and we got a dramatic improvement in kill rate. And you could use there was someone was using 1.5 litres at 360, which is a quarter rate, and getting almost as good a response as a full rate by itself. So the biology will have a really good impact in a lot of different areas. And where I'm getting to is that we need the guys like yourself and other consultants to sort of go, okay, like what would happen here or what would happen there. And another quick example is we looked at sap testing, and we put EM in with Wuxel brassica just in the glasshouse, and then we tested a month later, and the nutrients of the EM with Wuxel brassica were far higher, or the key nutrients that are in Wuxel brassica were far higher than Wuxel brassica by itself. So it's showing that it's adding it into the plant, and as I said, it's the mechanism through the stomal opening, and also if it gets into the soil, it's going to come through the roots. So biology has a really strong role to play, not just in the way that a lot of people think. Like people think when you're putting biological product on the soil, it's just going to, whether it be break down organic matter or change the soil structure or what have you, it can play a role in so many different areas.
Yeah, absolutely. So I guess if a farmer's getting curious about getting started on their biological journey, what advice would you give them?
I mean, first understand where you're at, I guess. Like, having a bit of insight into your particular farming system and what you're doing to impact biology or foster it in a lot of ways, and then go for it and test. Go grab a few paddocks. I often say grab your worst paddock, because they tend to be the ones where we see dramatic impact, because often they are carbon rich but just like function poor, and so where we can put a bit of function in there, we get a really good response. But like, start testing it out on paddocks, dig some holes, have a look, and it doesn't have to be a super expensive process to try out. There's so many different things. We have growers who are using using waste products in their area and using microbes to facilitate that to become a beneficial additive to their system, whether it be different manures, whether they've got like there's pig manure or chicken manure around the neighborhood, people with whether it be straw, whether you've got access to some sort of liquid effluent, and then utilizing biology to help supercharge that and then use it within their growing system, or just buying a product, whether it be a feeder, as I like to call them feeders, we're putting fish or humic acid or something, or seaweed, that I like to refer to those as feeders. So they feed the biology to try and kickstart the system, whereas we tend to be adding biology, or now with some of those other products, we're sort of trying to do both. You come at it from either angle, like we've seen it work all those ways. But have a crack, try it out, and then monitor it, and see what works, see what doesn't work, and then evolve, because that's what and just understand that we're working with a living ecosystem. So we're trying to foster and enhance that system, and boost diversity within that system. And if we can do that, you're going to see a huge amount of benefits and a huge amount of growth in your overall system and in yourself as a whole. I'll give a couple of quick examples. We had this grower who, this is years ago actually, I think one of the first times I met Nicole, she was speaking at one of these events, and we went out, it was out Rangiora way, and this guy had a huge pile of straw with manure through it. And so he had a huge shed. It was a couple of acres under roof, and in the winter he would winter his bulls in there, and essentially was putting down straw, and he'd muck it out and then just let it sit there, and it would just break down over time, and then he'd throw it in his maize paddock when he worked it up. And it was working fine, but I was there that day, and I just said to him, like, have you do you get complaints from your neighbour? And he was like, oh, actually my neighbour's a lawyer, and he has been looking at me a wee bit weird over the fence. And then I said, oh, look, I reckon we could do this a wee bit better. Why don't we try putting EM over the pile and covering it, and so essentially creating an anaerobic compost. And so we went and we put EM over top of it, and what that does is it doesn't allow that volatilization. So instead it changes it from a decomposition process to a fermentive process. So if you pile 100 tons on and then you throw EM on and cover it, you'll end up with 100 tons of fermented, what we call bokashi, or a fermented material that will break down rapidly within your soil and have a lot of energy, because it's holding all the energy. So when you don't have it covered, like it's a decomposition, like compost, you often see that all of the steam and all of the heat that you might, often on the news, compost piles burst into flames, because it's the energy, and that energy is going into to the environment, right? You're burning carbon, and the energy is going up, heat. And so we're keeping that in. And so he did that, and then so he had all this material, and so suddenly he's putting it on his maize, and he's also putting it on fodder beet, his fodder beet ground, so he had to weigh more. And then the next year he was sort of like, oh, why don't we try covering this in lime instead of covering it in a old silage cover? And I was like, oh, it might work. I don't know. So he gets a digger and covers it in lime, packs it in. At the end of the season, it worked perfectly. We were testing it because you can check temperature. So it'll ferment at sort of 35 degrees, rather than a compost heap sort of 75 to 80, really hot. So it's a good way to see. And then we did some testing, because we tested the pile before, and we were noticing that nutrients were way up. Its nitrogen was higher in the one that we covered, because we're not getting all that volatilization. And so the next year he was like, okay, this works great. He goes, now I don't want to spray EM onto it. What I'm going to do is I'm going to put it in in the cow's water, and they're going to be mobile applicators of this of EM. And is that going to happen? And I was skeptical, if I'm honest. Because I was like, how's that going? That might change the microbes, because it's going through the rumen. And so he, sure enough, he does it. He feeds it through them, and we get a perfect fermentation, and he gets a perfect product out the back end. So this was, I think I met him 2014, and he's still doing it now. It's been super cool. So we're able to see that the microbes, obviously, some of probably the yeast, your Saccharomyces are really good for animals, but that's probably given the animal benefit on the way through. Absolutely. But everything else has just come out the other end, and been spread perfectly and then help that fermentation. So it can sort of, and that's an example of a guy utilizing the biology and utilizing something else that's happening on his place, or a waste substance he's creating on his place, to massively benefit a system like those. And those soils that were going into beet and maize were obviously getting battered around a bit. They're pretty tough crops. You're in situ feeding beet, which is really hard on soil structure and biology. And then maize, it's a really hungry crop. You're putting a lot into it, but he's able to mitigate how much nitrogen he's putting in because he's getting this nitrogen-rich, carbon-rich fertilizer amendment. And then on the beet side, he's getting this really, really microbe-dense, beneficial, essentially compost that he's putting in to help to mitigate some of those issues that those crops create downstream. So I think, in a long way of answering your questions, we can really utilize other things on farm to pair with biology and some really innovative ways to really benefit your system and the whole diverse ecosystem of biology under the soil, and then also have really good downstream effects to help mitigate the managerial effects of farming, which are a reality in a lot of ways.
Yeah, absolutely. That's a fantastic example. I'd love to go have a look at that and see what he's up to. That's some pretty cool stuff, but it just shows you what's possible when you start, thinking about what can I do with this? It's not a waste product. How can I turn it into something that's really good? So I finish.
Yeah, exactly.
Yeah, I finish up all my podcasts with a quick fire question for everyone. And that is, I guess, what's one belief or principle that keeps you grounded no matter what's going on or what season you're in?
Like as I mentioned, like my background was commerce coming into this, and Mike's background was science, and that's why we work really well together. And he's sort of a one in a million guy that sort of let me do anything within the business and really let me grow. But one of the things is like I've been on the front lines and really had a huge learning curve with agronomy and biological systems, and now and I've been able to bounce things off him for the last 14 years, and now my knowledge probably rivals his in a lot of ways, and it's that knowledge and learning about the ecosystem that grounds me and gives me the belief that we're doing the right thing. We're like, this is going to benefit like boosting biological environments and creating system change within soil and waste ecosystems is a real big improvement that will be a really big improvement for humankind. And it's a really important evolution that we need to make, because we need to work with environmental and biological systems, not against it as possibly we have done in the past. And so I'm constantly reminded that and see the good in what we're doing and helping to benefit that, and that sort of keeps me grounded and keeps the motivation. And when I go to a grower and they're like a kid excited about seeing earthworms, or they've never seen clover like that, or they their crop's really healthy and they haven't used 2 fungicides that they were expecting to use, that stuff is just awesome. It's really cool and really keeps us sort of fired up for what we're doing and what we're trying to achieve.
Yeah, I love that. That's great. So if listeners want to, connect with you or learn more about EMs, where can they go?
Yeah, our website is EMNZ.com, and we're on all the platforms, so they can just touch base through one of those. And our website's got a heap of information. We've got like a heap of trial data, a heap of case studies, which are just sort of interesting things that come from really, really innovative growers that we start to figure out what EM does and how it can work. And then one of the cool things I started doing when I first came back into the business was Mike actually said, why don't you start writing articles? Because I sort of liked the web stuff as well back then. And he was like, it'd be a good way for you to learn. And we've sort of got like 400 articles on there now. And it's been an amazing way for me to learn about the product and how it works, but it's also a good resource for people if they want to find out something about how the product works or what it does. But yeah, just touch base. We can help out with anything.
Yeah, awesome. I've definitely got some more reading to go and do on your website by the sounds of it. There's a lot more applications I need to start incorporating it into and trialing it with some of my farmers. So I feel like I could have kept talking to you for another couple of hours and getting more into these case studies. But thank you very much for your time today, Paul. It's been great.
Yeah.
I've learned a lot, and
Maybe that'll be for the second time.
yeah, exactly. But yeah, I appreciate you taking the time to come on today and share a bit more about EMs and guest biology as well. I've really enjoyed the chat.
Yeah, no, thank you, and I appreciate, and I'm sure the listeners appreciate everything you're doing to give voice to topics that not a lot of people do in the market. So good on you.
Thank you.